Hi, I'm Hannah.
Hi, I'm Savannah. We decided to do our project on the anthropogenic effects on aquatic health. Essentially, we wanted to explore the relationship between how much the human destruction of vegetation surrounding a retention pond affects its aquatic health.
Having lived in Florida our whole lives, we see a considerable amount of retention ponds and we're curious about their aquatic ecosystems, so this project was perfect to satisfy our curiosity. A riparian is the surrounding vegetation of a body of water. A riparian can be beneficial to the aquatic ecosystem it surrounds, as it can help stabilize water temperature, dissolved oxygen, nitrate, and phosphate levels. Higher nitrate and phosphate levels lead to possible algal blooms. Algal blooms can lead to reduced dissolved oxygen levels, which can be extremely harmful to aquatic life. If dissolved oxygen levels drop too low, the retention pond may not be able to support aquatic organisms.
Various levels of anthropogenic destruction of riparian zones decreases biodiversity and negatively affects concentration levels of nitrates, phosphates, and dissolved oxygen. To test this hypothesis, retention ponds were found for each level of riparian length, and biodiversity and water health were measured. No riparian was defined as less than 0.6 meters, medium was between 0.6 and 2 meters, and full riparian was more than 2 meters.
A dip net was used to graze the edges of the retention ponds to pick up organisms in the water. When searching through what was picked up, each organism was documented, and tallies were used to account for finding several of the same one. Once accounted for, all organisms were placed into a bucket of pond water until all scoops were completed in order to avoid replacement. To ensure more accurate results, this process occurred at 5 randomly chosen areas. The accessible perimeter of each pond was sectioned off on a printout of the pond from Google Maps, and then was numbered. A random number generator selected 5 numbers, so one scoop took place at each of these 5 chosen locations. The water samples were taken at the first location. Test kits were used to determine phosphate, nitrate, and dissolved oxygen levels. These 2 images were examples of retention ponds used and how we randomly sampled for accurate results.
Results from the test kits showed that all the retention ponds, regardless of riparian length, had healthy levels of phosphates, nitrates, and dissolved oxygen. Phosphate levels range from 0 to 70 parts per billion, and phosphate levels under 100 parts per billion are considered healthy. Nitrate levels ranged from 1 to 3 parts per million, and nitrate levels under 4 parts per million are considered healthy. Dissolved oxygen levels range from 3 to 7 parts per million. Ideal dissolved oxygen levels are from 4 to 6 parts per million. However, aquatic life is well supported with dissolved oxygen levels between 3 parts per million and 7 parts per million. These images are examples of tests we used for one of the locations.
This chart shows the relative levels of each organism found at each location. There is clearly an overwhelming presence of the serrate crowned snail at the Florida orthopedic location, which will be addressed later on in this presentation. There were a variety of different snails and other invertebrates, as well as some vertebrates found.
The dark green represents the average Shannon Biodiversity Index values for each riparian level, including the data for the serrate crowned snail. The light green is the data without the serrate crowned snail. We use the Hutchison t-test to determine statistical difference between riparian levels. The background research strongly suggests that differing levels of riparian should have statistically significant effects on water health. However, our results showed that there was no statistical difference. This would mean that there is no relationship between riparian level and biodiversity.
These discrepancies could have been affected by several factors. Only the edges of the ponds were sampled with the dip net due to a lack of resources to safely test the middle of the ponds, so the results cannot be generalized to the whole pond, as there may have been many organisms overlooked. The levels defined as medium and full riparian in this research may not have been sufficient to have a measurable effect on water health. The test kits may not have been accurate enough for gathering necessary information. There may have been a combination of these factors as well as other factors not detailed here. Our research in determining anthropogenic effects on aquatic life is just the first step. While our results were inconclusive, more investigation should go into this issue. Understanding how human interference in the environment can impact ecosystems, even on a small scale, is important. Although this investigation did not support a correlation of riparian level and aquatic health, low aquatic health can pose a real issue because a cascade effect in the food chain would lead to decreased bird populations and increased bird prey populations. An increase in mosquito populations poses a threat to humans as they can transmit disease or trigger allergic reactions.
A snail was initially misidentified as an invasive species from New Zealand. After contacting the Florida Fish and Wildlife Conservation Commission and sending images to their biologist, the snail was identified as the serrate crown snail. This snail is very similar to the New Zealand mud snail, but it is indigenous to the U.S. The presence of this snail in such high numbers is negatively affecting biodiversity levels. The reason for the proportionally high populations of this snail, even though it is not invasive, remains a mystery. As seen in the organism chart from before, the presence of this snail is overwhelming. This image is of the serrate crown snails found at Florida Orthopedic. Finally, there happened to be a lot of trash at the locations we visited, so we cleaned it up. We appreciate your time.
Thank you.